Yeah, so thank you very much, and I'm happy to answer the question. First of all, I'm not the president of GZEF anymore. So GZEF, the German Society of Extracellular Vesicles, was founded in 2017 with some other scientists. And now on the latest MOVE meeting, we had our assembly and we decided to step down from the 5 who were in the original board to have some younger people, which now run GZEF with Kerstin Menk as the current president. The idea to the exchange was that we want to keep a young society, and we involved these people since a year to train them so that GZEF continues. And yeah, as a national society, when I was the president, we tried to interact with other national societies. And so there were some close contacts to the Spanish society, GIVEX, and to the British society, UK EV. And we discussed that it would be marvelous to exchange students from lab to lab and support their journeys. And yeah, we were planning that, and then we quickly received interest from other societies. That was the Austrian society, that was the Italian society, Evita, and it was, I think, the French society, FZEF. And so we were 6 societies and discussing then the exchange, and then it was COVID and we had to delay, and there were even more societies being interested, the Belgians, the Netherlands, and then we said, why don't we open it up, and that we have a platform in Europe where we, yeah, in a type of federalism, interconnect the different national societies or networks and interact, and to provide a scholarship of maximum 2,000 euros that one of the societies can be sent to another country to have this network idea. And so we had regular meetings, and then the program was kicked off, and we have the first MOVE students already ongoing. And since it's also good for networking to have meetings, and the International Society of Extracellular Vesicle meeting, the ISF meeting, was in Seattle, which is for many of the European young students not affordable, we discussed to have a meeting in Malaga. And that was also heading back to the early days when UKEV and GIVEX interacted together with Hernando del Portillo. He suggested, why don't we meet in autumn in Malaga? And I think it was planned for 2020 already, but then due to the pandemic, it was postponed. The MOVE activity grew. And so then we realized the meeting in Malaga this year. And yeah, and it was really a great meeting with around 400 participants. So it was a pleasure. Yes, so I'm very happy to do that. Maybe it's a little bit untypically, and maybe I don't know whether it's a good example how to do it. So I grew up in the area of Cologne. I studied in Cologne, and I did my PhD in developmental biologist working on the fruit fly. And instead of moving to the States for a postdoc period where I had some offers, I decided to stay at the area, so in my home area. So I had to change the topics, and after a short excursion to the hematology, oncology department in Cologne, I moved ahead to Düsseldorf and started to work with hematopoietic stem cells. And Düsseldorf is very close to Cologne, so it's easy to commute. Yeah, and there I started to work something to interconnect my Drosophila research with stem cell topic. And one question in stem cell biology was, how do stem cells self-renew? And one idea is that they divide asymmetrically, that one daughter cell retains the capability of the stem cell and the other gets specified. And this was exactly a process where I closely worked in that frame in Drosophila. And so Drosophila was the pioneering field. And there it was markers were identified which segregate asymmetrically during cell division. And so my mission at that time was to search for proteins which are inherited asymmetrically in dividing hematopoietic stem and progenitor cells. It was a long journey. We looked for cell polarity first, and finally we identified 4 marker proteins. And 2 of these marker proteins were tetraspanins, CD53 and CD63. And so then I started to read what are tetraspanins, and then I found, okay, they are membrane microdomain organizers, but they are also have been used for exosomal markers, and that was around 2005. And I appreciated the field, the idea of exosomes as complex extracellular organelles to transmit signals in between cells. And I enthusiastically read all the articles, but I said I cannot step into each field. And then for tracking asymmetric cell division in living cells, we thought it's a good idea to clone CD63 as EGFP fusion protein encoding region. And that is what we did. And then we started to transfect the first cells, and where we expectedly obtained 30 to 50% positive cells. And with the constructs encoding CD63 EGFP, we had 100% green cells, which was a surprise. And then immediately the idea came up whether now these successfully transfected cells secrete green labeled EVs, which are uptaken by the others, and therefore transmit the EGFP to the other cells. And then it was an easy experiment to spin down the supernatant by ultracentrifuge. And then we observed that there were numbers of green particles. And that was indeed the stepping into the EV field in 2009. Yeah, and so we were interconnected with stem cells, regenerative medicine. We had mesenchymal stem and stroma cells, we had endothelial cells. And now having been started, we were on a retreat and met people working with calcium phosphate nanoparticles, and they knew how big they are, how many they have. And I asked, how do you do that? And they explained they have a nanoparticle tracking device. And then I asked, do you think we can use it also for exosome quantification? And he said, yeah, give it a try. And that was indeed, we ran for it, and we published as one of the 2 groups in 2011 that NTA can be used as a tool for EV preparation characterization. And yeah, that was the step into that field. Yes, so I'm very happy to do that. So as I said, connected into the stem cells and having the NTA device and having mesenchymal stroma cells in the lab as stromal supporters for hematopoietic cells. And as we observed, they release EVs. And I'm becoming aware that MSCs act via the EVs by papers from Sai-Kang Lims and Giovanni Camusi's group. I became very interested in the MSCVs. And it was also 2011 when we had a patient of graft versus host disease, which was treatment refractory and should have been treated with MSCs. But unfortunately, a phase 3 clinical trial failed to show efficacy, so that the doctor could not use these MSCs, which he originally intended. And so we discussed about the MSCVs. These days we call them exosomes. And yeah, we discussed whether it could be feasible to treat the patient with the MSCVs, and we agreed we did. And it was a success. It became a landmark study. It was published in 2014. And if you look and observe parallel to the MSC field, we have learned that in the MSC field, also one of the first indications which was treated with the cells was also GVHD. And it was a long journey. And then there was a company, or is a company named Mesoblast, which translated the cells into a phase 3 clinical trial successfully, then published in 2020. And it was still refused for market authorization by the FDA with the argument there is a lot of product heterogeneity. And this heterogeneity aspect was also then, since many years, determine our research interest that and indeed we observe we have MSCV preparations which are potent and others which are not potent, having the same situation. And we searched for surrogate assays, functional assays, which can be used as potency assays. We have explored about that. And finally, we had to learn that even if you use with a given MSC stock, that there is always a chance to get active and non-active products. Sometimes it's 80% chance to get active products, sometimes only 20%. And resulting in the inconsistency. And to that end, we have the same situation than Mesoblast, that we cannot have consistent products. And so the question is, how can we overcome these limitations? How can we bypass this heterogeneity? And to that end, we decided to immortalize MSCs with a TERT-based method and some other IP-protected sites. And that works very well. And we expanded then single cells to have monoclonal cell lines. And we have demonstrated that these monoclonal cell line derived EVs are still potent in vitro and in 4 different animal models. And that is what we are currently using, that we work to better understand the so-called CI-MSCVs. We want to set up more functional assays. We have some good assays already established, but we think they act at different sites, and for each site we should have an assay if possible. And now we can scale the production procedure, and so my vision would be to become able to treat 10,000 of patients with MSCV products. And therefore we can not use the current methodology for upstream and downstream processing. And we need to think of how we can translate the production procedure to the industrial level. So having bioreactors, having other downstream processing methods. And the next milestone for me is to identify an appropriate cell culture media, which ideally is chemically defined and still allows secretion of potent EVs. Yeah, and as another area, you need to have quality control analysis, and to that end, Andre Gergens in the lab introduced imaging flow cytometry for the analysis of single EVs, which was massively extended by Tobias Stertel, and now Tobias is able to have in a medium throughput EV characterization at the single EV level from primary body liquids. So it was initially for the MSCV characterization, but now he stepped into the field of biomarker discovery, and we have started to publish, and there will be much more. Yeah, at first it was, for many years, we missed any functional assay which was qualified. And that means you had an MSCV preparation and you applied it into animals, and sometimes you were successful, sometimes not. it's said the process is the product, or at least the process defines the product. That means if you want to optimize methods, you need to have a good readout, which is qualified to give you the appropriate information about the potency. And we have to have also surrogate analysis or single EV analysis. And as I mentioned, we introduced NTA as one of the two groups, and we focused on NTA. And initially, we believe that each particles we analyze is an exosome, and evaluated purification methods according to the particle recovery. And then at a later stage, we learned this is completely wrong, which is massively puzzling also the field. That most of the particles in our preparations are not EVs, but other particles, and that we should not evaluate the methods based on particle recovery. There was a lot of failures. So it was good to have the imaging flow, which helped us now to improve the methods. So this heterogeneity is a big issue. So luckily we overcome it, as I elaborated before, with the MSCVs. And now the big challenge is the definition of the medium, as I said. For now, we use human platelet lysate as a supplement, which is expensive if you want to scale. It needs to come in GMP quality. We recently observed batch-to-batch variations, massively also affecting the product, which we did not consider that it is that intensive. And then the platelet lysate is very protein-rich, and the high protein contents challenge purification downstream methods. Specifically, we need to discuss how can we reduce volumes. So in the field, many groups use TFF, and we are using it also since more than 10 years now, and try to overcome the challenges of the protein-rich media. And it's really a massive challenge, and I think it's hardly possible. So the TFF works brilliantly if you have chemically defined media, but it does not work with 10% platelet lysate. And so what we wanted to do now is to get rid of the platelet lysate, and the first requirement is that the cell grow, and that is what we achieved. But now we learned that somehow something is missing in the currently tested chemically defined media, because the cells stop EV secretion, which we can analyze specifically because we have the imaging flow cytometry with the NTA and Western blot and other methods. You would not see that. Yeah, so this is the next challenge, and we have solved that. Yeah, and to do all this, we need money, and so getting enough funding, maybe this is the most challenging part. What we need to have, we may not necessarily go for large scale immediately. We should go for efficacy and for safety. And what we should be aware of is that the production process as such is robust. So at the moment, as said, we produce we expand the cells in 2D. We have the platelet lysate, which I don't like. We have some downstream preparation methods, which is pre-EG precipitation, which is ultracentrifugation. But now with the clonally modelized MSCs, we are able to have, with the same platelet lysate batch, consistent preparations, and in principle can translate that. What we should have in mind, how big is the patient group which we want to treat. So if it's a small cohort like a rare disease, or if it's just applied locally, and we can have with a scale out, that is meaning the same production process, you do it, amplify that procedure. If we can saturate the market, then it's time to go, and that is what we are planning. If you have a larger disease cohort, so we are discussing ischemic stroke together with Dirk Hermann. Of course, we have much more patient, and the scale out is too limited in scalability to reach to become able to manufacture for more patients. So that means there we need to go into the bioreactor and to the industrial level. That requires more time. But we urgently should, if we have a robust product where we are sure that the quality is great, it's urgently needed that the community start with some successful clinical trials. Otherwise, the whole field is losing trust. Yeah, at first, I think one should be curious. We should be enthusiastic. We need to like the research question, and it should be a component that is also a hobby. Yeah, it's not for people who have the work life or like the work life balance and think the life is other activities than research. It might get challenging, so you cannot do the job in 8 hours. You need to have much more passion, and of course, I also have other interests and spend my time. So we should have also some recreation, but yeah, it's you need to have fun and you need to be enthusiastic, otherwise it's not appropriate. And what I learned during my research, I was always a little bit rebellious and challenging things and challenging models. And I have learned we have still so many dogmata in science, that there are well established models around, and as a student regularly you believe them, and if you perform experiments which do not show the same results as a student and also as a supervisor, typically you say there is experimental misdesign, but maybe it's the model. Yeah, and one example is in hematopoiesis, where we did some good stuff. It said that hematopoietic stem cells create common myeloid and common lymphoid progenitors, and that myeloid and lymphoid lineages completely separated. It was challenged 2005 by the observation that there are some cells which do not fit into that model, and Andre Gergens dissected it in my group completely, and we saw that the neutrophils are born in the same branch than the lymphocytes, and not in the branch with erythrocyte, megakaryocytes, and the other granulocytes. And yeah, so still science is using very often this classical model, and yeah, having not the right model, you will not perform the right experiments. So remain critical, trust your own results if you are sure that you did everything correct. And yeah, so sometimes I feel back in the Middle Age, the Earth is a disk, and everybody knows the Earth is a disk, and it took years and decades until it was accepted that we are on a globe. And there are some dogmata around which I believe are very comparable and throwing sand in our eyes. Yeah, so to to address the challenges appropriately, that means to have enough money to continue with good research. And indeed, we want to go into the clinics so as soon as possible, as I said, with a scale out procedure. And we need to optimize a few final steps to have an yeah, a product which can be sufficient for maybe treating 100 patients a year with a strategy, and simultaneously work on a strategy to get able to do it in bioreactors, so to optimize the processing. This is something we can outsource to a company, and indeed we have founded a company named Exoslar, and we are now establishing a consortium of investors that we can outscale that. Then another important thing is potency testing, and basically with Sai-Keng Lim, I coordinated with other experts in the field an article about potency testing. How can we develop potency tests and that is we need to understand the mechanism of action, which is a little bit challenging. How can you do that? But this is somehow back to the roots to what my start in biology as a developmental biologist, where we dissected the notch signaling pathway, for example, in Drosophila melanogaster. So that means from my original education, I should be able to dissect the mechanisms of actions to have enough ideas. Now with the monoclonal cell lines, we can genetically engineer the cells, we can overexpress certain factors, we can knock down other factors, other genes by CRISPR-Cas technology, thanks to this discovery. And that means now we have started with genetic engineering of the clonal immortalized MSCs, and there we want to see, can we increase the potency, can we decrease it, but we can become also able to go back into cell biology and ask which pathways are required for the EV secretion, for example, if we interfere with the endosomal machinery, with the escort machinery, with metalloproteinases on the surface for signal transmission. And my vision would be that the production and clinical trials are outsourced to the company, basically, because you need another skills. And that then in the academic research, my group can continue to do more basic stuff and dissect the biology of the cells and understand more about the EV release and transduction machineries.